Ngā mihi mahana kia koutou katoa. Welcome everyone to this inaugural professorial lecture featuring Professor Logan Walker. My name's Tim Wilkinson and I'm the Deputy Dean of the University of Otago Christchurch, and it's my pleasure just to brief you on the order of things that are going to happen. So the order of events is that in a minute Logan and the academic party will come in, and then Associate Professor Justine Camp, who's our new Associate Pro-Vice Chancellor Māori in the Division of Health Sciences, will welcome us more formally, and then she'll pass the baton, shall we say, to Martin Kennedy, who's HOD for Logan, and he'll introduce Logan, and then Logan will do his thing, which we'll look forward to. And then the Dean of Christchurch campus, Suzanne Peterma, and who's also our new Dean of the Otago Medical School. So that's the sequence of events. Probably take about, I don't know, well, we'll see. It'll take as long as it takes, probably about an hour or so. We'll definitely look forward to it. So I had to look it up, but I thought, what is an inaugural professorial lecture? And it is, of course, to celebrate people who've recently been promoted to professor. And the aim is to celebrate and promote the work, the academic work of Logan. So we're definitely going to look forward to that. Can I ask you to stand, and we'll ask the academic party to come in.
On in and everyone stands there for an hour. Sorry about that, guys. Awa tēnei te ruru e koukou mai nei ki hai mai hiti hiti ki hai marakaraka. Te upoko nui o te ruru te rukau he pō he ao ka oatia. Tēnā tātou katoa, nau mā hara mai ki tēnei whiti kōrero ki tēnei hui whakahira kei o tākou whakaihu waka kei o tautahi. Ki oku whanaunga no tuatahiwi, te murumuru o te ahi o konei, tēnā tātou. Ki a koe Suze, koutou koto tīma tino pai. Naui whakatere tēnei waka, koutou koto tīma hei whakakōkiri i ka mahi katoa tēnā koutou. Ki a koe Logan, ko koe te taki o te pōnei. Ki tō whānau hoki, Margaret and Stuart. Rachel, Sherilyn, Dean and Zoe, and other whānau members, welcome. Kia koutou katoa, e hara mai ki te whakarongo ki tēnei kōrero papairau atu nau mā hara mai. So welcome to Otago Whakaihu Waka, University of Otago, Otautahi Christchurch. For this very special occasion, the inaugural professorial lecture for Professor Logan Walker. We are incredibly proud to host this event at Otago Whakaihu Waka, a place dedicated to excellence in research, teaching and innovation. This is not only a significant milestone in Logan's career, but also a proud moment for Otago Whakaihu Waka and the University of Otago, and especially the Christchurch campus. Thank you all for joining us this evening to celebrate this achievement. We hope you enjoy the lecture and feel inspired by the insights and discoveries that will be shared tonight. I now wish to invite Professor Martin Kennedy to introduce Professor Walker. Nō reira, tēnā koutou.
Kia ora, and thanks to Professor Camp for opening the proceedings. As Justine said, I'm Professor and Head of Department of the Pathology and Biomedical Science Department here at University of Otago Christchurch, and it's been my enormous pleasure to have had Logan as a member of this department for my tenure, but actually David Murdoch reminded us a little earlier this evening that he he was the original head of department when Logan actually arrived on the campus, and so there's a really nice connection there. So it's my job to set the scene a little bit, provide a little bit of biographical detail about Logan, and I have to say that's a huge pleasure for me because he's a friend and a close colleague for the best part of 3 decades, and we also have a shared interest and love of genetics, and so we have even more reason to collaborate and work together, and certainly have enjoyed Logan's company over the many years he's been with us. So I thought I'd start by talking a little bit about Logan's academic background. It's pretty important at an inaugural professorial lecture. Logan went to high school in Ellesmere College in Leaston, I believe. He grew up in the country on his parents' farm, and then proceeded from Ellesmere College through to University of Canterbury, where he did a BSc, followed by a Masters of Science, finishing or graduating from that in 1997. And at that point, Logan came over to the University of Otago Christchurch, which was then probably called Christchurch School of Medicine or something similar. And he started working as a sort of junior researcher and progressed from there, enrolling in a PhD with the late Christine Morris, Dr. Christine Morris, who sadly passed away last year. And so Logan worked with Christine on cancer genetics, completing his PhD in 2007. And pretty soon after that, he uprooted his family. I think they were probably willing accomplices, I'm not sure, and moved to Brisbane in Australia. And he joined the laboratory of Professor Amanda Spurtle at the Queensland Institute of Medical Research, Berghofer, which is one of the premier medical research institutes in Australia, very strong in genetics, very strong in cancer and many areas. And so he did a lot of his postdoctoral training there. And I know after 4 years he was looking at returning home, and I know that Mandy Spurtle was not at all happy that Logan was leaving. She was very reluctant to let him come back to Christchurch, but we were very glad that Logan's prevailed and came back and joined us. He returned to Christchurch and joined the Mackenzie Cancer Research Group here in 2011 as a research fellow, and started the journey that he's now well along, having just gained his professorship. So it's just a few words about Logan's research. He's obviously, as I've said, an expert in cancer genetics. He's got a very strong international as well as national reputation. He's either chaired or is a member of many international cancer research consortia, and these groups tend to work on the gene variants that influence risk of cancers. And I'm sure Logan will say more about this, and probably more eloquently too. And rather uniquely, Logan's channeled those international connections which enable both New Zealand researchers and clinicians to interact with international bodies and get the latest information. And I think his research has a translational element that I think has really helped with cancer treatments in this country, or the understanding of cancer at any rate. And he's managed to also engage a team of PhD, postgraduate students and young researchers and mentored them through. And so they get the opportunity to work on these large data sets in the company of many international experts in this field. Logan's published 70 papers or so, give or take a few, in scientific journals, international journals, which is a very impressive output, and these have been cited over 2,000 times by other authors, which gives you an idea of how well received and important the research is. Logan's also given 30 international invited talks on his research, and countless national talks and local talks. He's a prolific speaker, often called in to talk at conferences and seminars and so on. But all of his research has been fueled by his ability to attract research grants and research funding, which is very challenging. But during his career, he's won over $7 million in research grants from places like the Health Research Council, Cancer Society, other cancer charities, and Canterbury Medical Research Foundation, and so on. And he's also won another $3 million as a co-applicant on grants. And I think most notably, he's also won the two most competitive and important research fellowships that this country has on offer. One is the Sir Charles Hercus Research Fellowship, and the other is the Rutherford Discovery Fellowship. And he's won both of them, which I think is pretty unusual. And in 2023, he won a University of Otago Christchurch permanent fellowship, which is also a very rare thing and wonderful thing. And so we're very pleased that Logan is now secured in his career. He's also a popular teacher and a supervisor of postgraduate research students. I mentioned some of his students, but he supervised something like 23 PhD, masters or honours students. There's a long list of awards I'm not going to go over, and I will just say a few words and sort of to wrap up about service activities that Logan's been engaged in the sort of academic community. Expected to do research and teaching. Some of our staff tend to be more focused on research, but also you're expected to do service, which is the work you do for both for the community, the wider community, but also for your research community, for the university, and for that sort of fills the gaps and allows your expertise to be used in productive ways. So Logan's played many significant service roles. He always steps up in these things. He doesn't hide, and he's been on many grant assessing committees. He's examined many theses, many postgraduate theses, does a lot of peer reviewing of papers, sitting on committees and advisory groups and so on. Again, his expertise is pretty sought after, and he's a very collegial and effective member of these groups. I'd also point out that since 2022, he's been our Associate Dean of Research here at the University of Otago Christchurch, and that is a very big role, and he's fulfilling that role really well. In addition, since 2021, he's been the co-director of a University Research Centre called Genetics Otago with Associate Prof Louise Bicknell, who's come up from Dunedin. So Louise and Logan have run this very large grouping of geneticists in the university. And actually, and one other thing I'll mention is that he's very generous with his time. He's spent time with students, school students, and talking to public groups and so on. But one of his major contributions has been to Breast Cancer Cure's Fashion for Cure event. And I think that's where his wit, warmth and humility have made him a real crowd favourite at these large fundraising events. And I think he's done something like 10 of them over the years. There's actually a lot more I could say, but I really should leave some time for Logan. It's really his night, but I just would end by saying, well done, Logan. It's a huge step to becoming a professor. The university puts up a very high bar. You've made it. We're glad you have. We knew you would. And it's my real pleasure to introduce your talk, which is Preparing for the Future of Genetic Health.
Kia ora. Thanks, Martin. Tēnā koutou katoa. Tēnā koutou in rangatira. Nā hua mahi, tōku whānau. Sorry, just getting a little bit emotional. Mea koutou katoa, kua hui mai nei, nā mihi mahana kia koutou. Thank you so much for coming along and listening to me present this lecture. It's an honour to be here. It's an honour to work with people, and I'm not going to focus on the thank yous tonight because I'm a bit of a wuss. So we're just going to go through the lecture as planned and do it in a nice stoic manner. So yes, thank you so much for the introductions, and thank you to those who were in my academic party. Very meaningful to have that support. So I've entitled this Preparing for the Future of Genomic Medicine, and this is, I guess, a bit of a summary of what I've done in the past and where we're at now, and maybe a little bit where I'm going. But science, I wanted to be a scientist for a long time, But it wasn't my only career path, and as Martin said, I grew up on a farm in the country in Dunsandel, and that's a photo, of course. I'm the one on the left, and you know, many memories of the farm, and you know, one which I've talked about before, just in the family, but spent a lot of time on the back of the truck. Dad used to farm with his brother, and they would sit in the cab, and they needed someone to open and shut the gates, so you know, I'd be on and off all the time, and I'd be on the back with the dogs. And but on wet days, things were different. The dogs got to ride in the cab, and I stayed on the back of the truck, opening and shutting gates. So farming wasn't for me. I love that sort of lifestyle, but I wasn't a farmer, and I think I probably saved Dad a number of heartaches in that by going in a different direction. So, as Martin said, I went to Ellesmere College and then went to University of Canterbury to do my BSc and MSc. My Masters was looking at zebrafish and trying to develop transgenic zebrafish to grow large and so on, relatively unsuccessfully. But from then on, I got my first job here at the University of Otago in Christchurch, or what it was previously known as. And here's a photo of a group which was several labs. In this photo, you'll recognise the handsome chap on the top left. But this was around 1998 that I joined with Dr. Peter Cross, and he gave me a position. It was a 2-year position, and working with him, looking at non-Hodgkin's lymphoma. And this was a project where we were examining a DNA break that had occurred. You can see a karyotype. These are chromosomes from somebody with non-Hodgkin lymphoma. And Peter was successful writing a grant to look at the translocation between chromosome 2 with chromosome 14. And the important part of this was to look at the breakpoint and see what was going on there and what genes might be involved, because a lot was not known about the etiology of non-Hodgkin's lymphoma. So this was my 2-year, which was successful. We mapped that breakpoint and published a paper, but Peter then wasn't successful with further funding, and he decided that's it for him, he's going to retire. So I was out of a job, and I went away working in an English language school, just doing dog's body work, but that's where my wife was working at the time. So I kept myself busy. But in the meantime, Dr. Christine Morris, had written a grant, and she was successful. And so it was about 6 or so months later I came back, much to the disappointment of the lab, who had already given me a going away present, and here I was back in the lab, and they all demanded it back again, which they didn't get. So, Dr. Christine Morris unfortunately passed away last year, but she was my primary supervisor. The job turned into a PhD, and it was a PhD that I was able to do part-time while working. And so Christine partnered up with Professor Bridget Robinson, and Professor Bridget Robinson is recently retired, was a senior oncologist here at the hospital. And my project was looking at genetic aberrations in breast cancer, and so this was focusing on DNA within the tumours in the breast. And one of the key aims was to try and identify those early genetic changes which occur and drive breast cancer. So this was my project, and initially we were looking at core biopsies that were taken from women, going through a process of diagnosing whether or not they have cancer and what sort of cancer they might have. This was partnering with Hei Taonga Tapu Society Tissue Bank, where we get these core biopsies, and you can see in the bottom left, this is a picture of a core biopsy which has been taken from a breast, which contains tumour tissue and fat tissue and so on. And from that I extracted the DNA and then carried out a technology which was relatively new at the time, called comparative genomic hybridization. And this was a technology, again, we're looking at chromosomes, but this technology used DNA that we get from tumours, and it looked at DNA from normal cells and it labelled the tumour DNA, I labelled the tumour DNA green, the normal DNA red, and we co-hybridised it on chromosomes that were on a microscope slide. And then there was also a blue stain that was added to help us distinguish the different chromosomes apart. But you can see the different types of colours that were on the chromosomes, and using this I was able to tell where the large chromosomal changes were occurring. So we're looking at copy number changes, deletions, duplications of genetic material. And so, as many of my colleagues know, I'm colourblind. This is doing colour-based studies is not ideal, but for this, thankfully, we do have computational ways to analyse it. So it wasn't reliant on me just looking down to see whether it was green or red, or I don't know what colour those things are looked at. But I did have to learn how to karyotype chromosomes. So that was I really enjoyed doing that and developing profiles. And just before I move on, there were a number of other projects. There was another fluorescent-based projects which I really enjoyed. The pictures looked great, but I often had to get people to help analyse because there were some things that I just couldn't see down the microscope. So had to have very colour competent people support me with my PhD. But anyway, we got through it. And but one of the projects that we did, this wasn't actually part of my PhD, but this was from a summer studentship that we had years ago, and it was Hamish Campbell who joined me in the lab, and Hamish is now the MP for Ilam, but he carried on after this to do his PhD and work in research. So Hamish came in to work on a project with myself and a number of oncologists, and you can see Christine Morris' name there as well. And this was a family that had a platelet disorder and a predisposition to acute myeloid leukemia. And we looked at the RUNX1 gene to try and find what we called at the time a mutation, which we now call a pathogenic variant. And you can see the family pedigree here with these black indicating that they've either got a myelodysplastic syndrome or AML. And this is, for those who haven't done genetics before, this is a gel which we use. We're actually analysing DNA here, and in this situation you can just look down under each individual of the family, and what you'll notice is that the individuals that have a black in their circles are women, and the squares are men, and you can see that the profile looks a little bit different, and what we had identified and we were genotyping was a mutation that was found in RUNX1. So that was a neat project for a summer student, and we got a nice paper out of it. So, but that was my first introduction really to that type of genetics in terms of predisposition. After finishing my PhD, as Martin said, I went over to Brisbane and got a job at the QIMR Berghofer Medical Research Institute. Recently changed its name. It was just QIMR, which is the Queensland Institute of Medical Research, but Clive Berghofer gave them an inordinate amount of money, and they now changed their name. So if it was spelt out, it would be Queensland Institute of Medical Research, Berghofer Medical Research Institute. So anyway, very functional, and I was fortunate to have a wonderful boss, very stressful at the time. Mandy is incredibly talented, and I felt I had to lift my game significantly, and that was quite a stressful time actually in Australia. And I did all the wrong things when I went for my postdoc. You're supposed to do your homework and talk to people in the lab and see what these people are like, and I knew that I was losing my job here, the second time losing the job at the University of Otago Christchurch, and I needed to do something about it. And I talked to a couple of researchers in Australia, Mandy being one of them, and eventually they offered me a position. And what came of that is, that time Mandy has become a mentor to me, and oh gosh, now I'm even kidding. So really appreciated her help over the years. I meet with her at least once a week on Zoom, and multiple times in conferences and so on. So with that, I came back to Christchurch just after the earthquake and joined the Mackenzie Cancer Research Group, which wasn't called that at the time, but it was soon after. And this is a snapshot in time. It never captures everybody. This was taken a couple of years ago, I think. And you can see Bridget Robinson, who was my PhD supervisor, and Bridget and I had maintained contact, and when coming back, before I came back, I met with David Murdoch, who was the HOD at the time, and asked, if I got funding, would I be allowed to come back to the department? And David very graciously said, yes, that would be fine. So, and suggested I work with Bridget and in that group. So that's exactly what I did. So there's a number of faces people that I work really closely with in the group who weren't in this photo. I just want to acknowledge John Pearson, who, although not part of the research group, a number of the grants that I do, I work really closely with John. He's a biostatistician, bioinformatician, an expert in genetics, and we got introduced to one another soon after I got back, and we've been very close since. I just want to acknowledge, before I move on, talking about research, a number of the funding agencies that have supported my research, and you can see our group name called Mackenzie Cancer Research Group, and it's the reason why we call ourselves that is since 2010 it's been supported by a significant amount of money from the Mackenzie Charitable Foundation, which has been immensely useful for stabilising careers in the very, very difficult environment that we live. So before I go on to the research, Martin mentioned service, and I want to acknowledge that too. One of the, Louise Bicknell and I co-direct Genetics Otago, which is a University of Otago research centre which supports more than 300-400 genetics staff and students. We have a range of activities from supporting Māori researchers through conferences, outreach into schools and kura and so on. And there's a number of activities that go on, and I think Louise would both say without Rebecca we would only be half the centre that we are. So Rebecca manages the centre, and recently we've been joined by co-deputy directors Dr. Megan Leask and Associate Professor Michelle Knapp. One of my activities as the Associate Dean of Research is to run the UOC Future Health Researcher Program, which the Deanery developed, and this I just want to acknowledge because this does take a significant amount of time, and this was something which ably supported by Katrina Notton, who really over the last couple of years I do less and less. Katrina is an administrator here who, I don't know if she's in the audience, but she has looked after this program significantly, and under Dr. Annika Seddon, who has we took on as the director of the program, and between those two they've done a fantastic job of supporting our students over the summer. As ADR, I also get to chair the UOC Research Committee, so I have the privilege of working with this group of people. This is there's a couple of changes this year to the committee, but this is something which we meet regularly every couple of months and discuss research strategies and initiatives on campus and what's happening with the university and wider, and very appreciative for the support that these people give. And also I have the privilege of co-chairing with Dr. Christina McKercher, who's our Associate Dean Māori. We co-chair the Māori Health Advancement Research Committee, and this is again a privilege to mix with people who I probably wouldn't otherwise see quite so much of in my research only capacity. My children decided to, when they heard that I'd been promoted, they went down and bought me a plaque for the desk, and you get what you pay for, I guess. Which I love. And here we have mistakes in this case doesn't have a consequence, but spelling mistakes in our DNA code does. So it's a segue into thinking about genetics now. And the way that I'm framing this is to think about our clinical pathway that generally people would face in New Zealand. And then alongside that, I'll show you how our research aligns and hopefully impacts these different areas. So for genetic health services, a patient would be referred to genetic services, maybe from oncologist, maybe from GP, so on. And there will be an assessment, and it might be the genetic counsellor or the clinical geneticist might determine that they should undergo genetic testing. And in such cases, blood, possibly sputum, might be spit might be used to extract DNA and then undergo genetic testing. And then the results from that genetic test come back, and finally that provides information, and it helps the clinician understand how appropriate management strategies should be undertaken, and for diagnoses and so on. So it's it's very easy when you put things in this in this type of pathway, but when we're looking at referring patients to genetic services, the reason why we do that is because people in our community have all different risks of developing all different diseases. And a lot of my research has been focused on different types of cancer, particularly breast and ovarian cancer. And we know that some individuals in the community who carry a genetic change, a genetic spelling mistake, if you like, that their risk has gone from what might be 1 out of 9 chance to get cancer in a lifetime to something that's closer to 8 out of 9. So gone from unlikely to develop breast cancer to very likely to develop breast cancer. And it's similar for other types of cancers and diseases. So we want to identify who those individuals are. A number of those people will turn up in oncology ward, and if there's no indication, maybe they don't have a strong family history, that there is that they carry a genetic risk, they'll be treated for the disease at hand, and hopefully successfully, and they will go home. But they go home not knowing that they're at really high risk of coming back again because they have a cancer predisposition. Pathogenic variant. So we want to establish how we're doing in New Zealand, how are we doing at finding these individuals. And one of the projects we were able to do with that was led by Dr. Vanessa Lau, who did a PhD with me and has stayed on as a postdoc and now a research fellow. Conducted a study looking at women who had been diagnosed with breast cancer between 2014 and 2017 here at Christchurch Hospital. And there was, I think, about 370 women who had been diagnosed with breast cancer and had consented to having blood and tissue banked at the Hei Tapu Cancer Society Tissue Bank, which has been so important for the research that I've done and others have done here at the University of Otago Christchurch. So what Vanessa did was look at the DNA, look at those genes which are cancer predisposition genes or breast cancer predisposition genes, and determine how many women carried high risk pathogenic variants, and then compared this working with our genetic counsellors at Genetic Health Service. Identify which women are at high risk, how many of those women, when they were seen with breast cancer, went on to getting a genetic test, and if they didn't, why didn't they? What was the reason? And what our study showed was about 50% of the women who were at high risk of developing breast cancer or ovarian cancer didn't have a genetic test, so they didn't, and there were various reasons for that, but it shows that we're not picking up all those individuals who are at high risk. And we talk about inequity in health. This is one of the highest inequities, when somebody is on your doorstep and they're at high risk of coming back again, and we don't have a mechanism for detecting it. We do have a mechanism, but of course to do that requires resourcing our genetic health service far more. So this was very consistent with what other countries were seeing, so I guess you can take a little bit of comfort for that, but it suggests that we're not doing enough in this space. The studies that I'll talk about next sort of bring into the huge international resources that we've developed, and this is one of the benefits that I had while working with Amanda Spurtle. She was a master networker, and because of that, I got mentored into a huge number of spaces. And so the symbols dotted across the map here are cities or countries which have one or more study sites that we collaborate with through a number of different international consortia. And for some of these, you'll see that South America and Africa and even some of Asia are sort of not well represented, but there are projects now underway which are starting to realise that there are underrepresented populations and we need to do something about that. So truly is a global international resource that we've, I was going to say tapped into, but we are collaborating. And a number of the projects, so I mentioned about sitting on the truck, you know, some of these consortia, you can just sit on the back of the truck and you can let someone else steer, but there are opportunities to sit in the cab and even drive if you want. And the projects that I've listed here, the study 1, 2 and 3, are projects that we've led here in Christchurch, but have involved many study sites around the world. The first being an Australian study, which was one of those situations where I talked about a project at a consortium meeting, and one collaborator came up and said, "Hey, look, we've got these genetic data. Would you be willing to do a study?" And we said, "Sure. You know, you're giving us something for free. You know, it's just our time." So we conducted a study, and you know, we've published a number of papers from these types of studies. But a couple of things I want to point out. One is you can see, you know, consortium, very important. You can see the diseases that we've looked at. Breast and ovarian, as I mentioned before, has been a very common disease that we've focused on. You can also see they're in chronological order. You can see how the study cohort size has just increased over the years. And you know, we're starting a number of years ago, you're doing cohort studies of hundreds of individuals. And now, you know, you can see this Confluence project, which is actually a consortia of consortia, now has, you know, hundreds of thousands of cases and controls. And I'm not going to talk about how we do these studies, but they're big genetic association studies. And what we find from these studies is new genes, new genetic regions which are associated with disease risk. The study 4 is one that's underway at the moment, so no publications from that, but really keen to see the data and see how we can lead in a you know, another project. So that with the breast and ovarian cancer. We've also been working with endometrial cancer, and again, these studies are in chronological order. The first study that we did, really collaborating with the UK and Australian study sites, looking at just under 2,000 individuals, and now it's you know, order of magnitude greater. And what these studies are also identifying is regions of the DNA that are associated with increased or decreased risk of developing endometrial cancer. So I wasn't going to go into all the details, but just to let you know the sorts of studies that we've undertaken in this space. I also want to, this is a little bit left field, but a while ago, Dr. Arthur Morley-Bunker came to me. He was he was a medical laboratory scientist working here in Canterbury Health Labs, and it was a cytology and a section lead, and he wanted to do a PhD and start getting more into research. And we started talking, and both of us had significant interest in digital pathology. And it's an area which I had a lot of interest in. I tried to get, working with people from the University of Queensland, get projects off the ground before coming back from Brisbane, but we weren't successful. And but this was an area Arthur was keen to go down. So he did undertook a PhD with me. And then later on, Dr. Andrew Davison from the UC, I had the privilege of co-supervising him. He came from a more data image analysis software sort of expertise, and between Arthur, myself, and Andrew and George and others from the lab, we have developed a series of projects looking at digital pathology, and these projects now what Arthur is leading, and I sit on the periphery and try and say supportive things and try and understand a lot of what's happening, because this is not genetics, but it is used to try and understand what's happening in the cells. And if what's happening in the cells is driven by genetic changes, then the question is, can we predict certain genetic changes by looking at cell morphology and so on? So digital pathology is trying to get what we'd normally see down the microscope, put it onto a screen. You can analyse the image in multiple ways, and including using artificial intelligence and everything that goes with that. So this is really going outside my area now, but it's something which Arthur is pursuing, and it's exciting to see where this might go. So this is still linked in with genetics, because there's still not everything they do is looking at genetic changes, but some of it is. So that's looking at trying to develop resources of understanding. If we're going to understand who is at high risk, we've got to understand all the genetic changes that might confer high risk, and we've really got to put into practice our knowledge and technology so that we can identify those individuals, with the hope of that we can prevent them from developing disease in the first place. The next step of not looking at the time here. The next step is to look at the test results. So it's very easy to send DNA away to get the genetic code read and it come back, and we can see AC, GT, all those sorts of things on a bit of paper. But we have to understand what it all means, and we have to understand whether a gene with a spelling mistake is actually going to cause disease. And this is really, really hard, and we're getting good at it for some genes, but there's much to learn. There's a lot of genes we don't understand very much. A lot of the work that I've been doing alongside is helping develop testing guidelines, and as a result of that, because of the work that we've done, we now start getting labs and various clinicians and asking us what do these results mean. It's come back as a variant of unknown clinical significance. Can you do anything with that? And it's one of those weird areas of science that I find myself doing, offering advice to labs. I don't have any accreditation. I'm not a clinician, and I'm certainly not getting paid to do this. But here you are with people asking you to do, and I think we've provided support for hundreds of genetic reports, and I don't do it by myself. I work with Mandy and other people, and it's incredibly rewarding because you're learning all the time, but it's really hard to manage time. One of the areas which oversees the genetic testing is this outfit called ClinGen. It's an NIH-funded. Not sure if it's funded at the moment in America, but ClinGen is it oversees the governance of the FDA approved framework for how to interpret genetic variants. And the purpose of ClinGen is really to bring together patients, clinicians, diagnostic labs and researchers. They want to aggregate all the data out there. Curation is deciding whether or not genetic changes are associated with disease or disease risk or not, or whether it's benign. How do we disseminate that knowledge? And of course, the ultimate goal is to improve patient care through genomic medicine. ClinGen have a number of working groups. They have a number of expert panels, which they have gene curation expert panels, which are trying to establish whether or not a gene is associated with a particular disease. They have variant curation expert panels, which is where I sort of fit in, is we know this gene is associated with a particular disease, but how do we handle all the variants in those that get discovered? So there are expert panels which sit down and they decide on rules which are long and lengthy, and other people have to read them, which is, feel so sorry for them. But here's a couple of expert panels. One sort of deals with BRCA1, BRCA2, breast cancer susceptibility genes. Angelina Jolie made BRCA1 famous a wee while ago. And then there was another cancer-associated expert panel, and you can see that as we bring out the rules, we publish them. So that's for BRCA1 and 2, and for this one we've done an ATM, and there's another gene, PALB2, and so on. So these papers are going to keep popping out as we share these rules. Along with that, ClinGen, one of the areas that we have to understand how to interpret rules is using lab-based functional experimental data. We can use population data, we can use computational, we can do a whole lot of different analyses to analyse this one variant that we've found in a gene. One of those areas is RNA diagnostics, and RNA is a molecule within the cell. It's like a photocopy of the gene, but one of the ways that we can determine whether or not a genetic change is causing a functional disruption is to look at what's happening to the RNA. And it's a very relatively simple procedure to do, but there's a lot of nuances that are surrounding it, and a lot of labs are using it, and a lot of heterogeneity of how they're interpreting what to do. And so there was a call to say we actually need to try and standardise how we're doing this diagnostically. So they pulled together, so ClinGen wanted to have a ClinGen, SVI stands for sequence variant interpretation, and then they wanted to have a splicing subgroup. So this is RNA splicing, it's a mechanism within the cell. They pulled together a splicing subgroup, and I had the fortune, because of the work that I'd done in the other expert panels in the area of splicing, I was asked to be on the subgroup, and there was 8 of us on this group, and our role was to develop a guideline that would then support more than 70 expert panels. So I haven't put faces, I've just put down the institutes that, so Broad, QIMR, because Mandy's on this panel as well, Natera, GeneDx, Ambry Genetics. So Natera, GeneDx and Ambry Genetics are big genetic testing companies, and little old University of Otago. And so we published a paper a couple of years ago, and this paper never had so many requests. And this is when it comes down, as soon as we did this, people just started firing, said, love what you did, you know, it's been really, really helpful, but we've got all these hard examples which fit in the gaps. And so suddenly we're presented with all the difficult cases and having to work out how we deal with these, which again is fun, but it takes a lot of time. And yeah, so we were really encouraged by what we did, and we're trying to think we need to expand this framework. So that's taken a lot of my time very recently. So finally, just want to talk about the personalised healthcare. So there's a number of actionabilities, I guess, that someone can take if someone is at high risk of breast cancer, for example. A very effective, a radical way of reducing risk is to have prophylactic mastectomy. And there are other ways, you know, you could have more intensive screening and so on for a range of different diseases and so on. But we don't do very well with prevention in this place. We have a lot of effort for early detection through screening, looking at diagnosis, prognosis, treatment, but we don't have a lot of investment in personalised healthcare where we've identified somebody at high risk. What are their options? How do we stop them getting a disease that they're predisposed to developing? And I've just mentioned refining risk assessment measures, which I'm not going to talk about here, but that's another area as we understand more about the genetics and other genetic modifiers on those genetics and the environmental interplay, all of that can modify risk, and assessment models are sort of being developed all the time. But I just want to finish by talking about novel risk-reducing preventative therapies. So this is work which is being led by Dr. George Wiggins and Dr. Vanessa Lau, and they've kind of gone in different directions, and I'd love it if they both worked. And so George has gone in a direction where looking at genetic markers, where we're comparing cases versus controls. We're looking to see what markers are more prevalent in cases compared to controls, and then determining, is that marker associated with decreased risk or increased risk, and so on. Can we use those genes that we identify as targets for preventing disease? And this sort of spawned by a large study that we did, which was actually led by Chris Hackart, who's in the audience as well, where we identified a gene which was associated with decreased risk of breast cancer, and George has taken that a step further to determine whether or not he can target this gene with particular drugs. Conversely, Vanessa's gone in a slightly different direction, using what we understand about splicing, RNA splicing, which I just talked about, to see whether we can modify this and actually manipulate it within the cell so that it actually bypasses the spelling mistake. And so Vanessa's been working on this, and both studies are relatively early, and I should say George has gone outside of there are other sort of screening methods which he's using, and Vanessa is very early into this work, and we've got high hopes. It would be fantastic if we started identifying new ways to prevent cancer. So I was just going to finish with this slide with genetic health and the cancer care continuum. And I mentioned before that we have a lot of work goes into looking at early detection, diagnosis, treatment. We don't have a lot in prevention other than population health, which is incredibly important. But when somebody is identified as having a genetic variant which has made their risk from relatively low to really, really high, you need something which is going to impact that significantly. And I just wanted to highlight that genetics plays a key role in each of these steps. We've talked about the idea of precision health, I guess, and the idea that if you're a low risk or a high risk, how do we treat or manage those individuals? What about prevention? What are we doing to develop new types of prevention? Genetics, our understanding of genetics can help us there significantly. For early detection, if we know someone's at high risk, what sort of screening methods, what can we do if we can't prevent getting the disease really, really early so that it can be treated? And then diagnosis. Genetics, of course, has a massive role in identifying, classifying different types of diseases and so on. And of course, treatment. There's a big push at the moment on personalised treatment, identifying what's happening in the tumour or a genetic change which someone has inherited. That's going to really determine how a doctor is going to treat a patient. So genetics is important, and this is an area where, I think it's a revolution has kind of happened, but the technology is just bursting, and everybody is going to be confronted with genetic health in some way at some point in the future because of what it's doing in cancer and outside of cancer. That's all I have to say. So thank you very much for listening. Thank you for everyone who's played a role in my career, and I hope over the time I have shown my appreciation for people here, and if I haven't, then shame on me, but really appreciate working here. One of the reasons why I wanted to come back to Christchurch after working in Brisbane is because I knew the people here and I wanted to work with them, and that's the scientists, administrative staff, and so on. It was just a really good culture in this place, and I'm very, very lucky to be here, so thank you.
Kia ora, Logan. You have now passed officially your last academic test. It's all just fun from here, I promise. I really wanted to talk actually to Logan's whānau because I get to talk to lots of other people in this room all the time. I just wanted to tell you what a privilege it is for us to have Logan on our campus. I think here at the Otago Whakaihuaka ki o Tautahi, we have what we call a po, which is, if you go into wharenui, it's like a beam that supports the whare, that holds it up. And I think in lots of ways, really, Logan is our po manawa here at the med school. I think you'd be pressed to find anyone that could say anything that wasn't positive about your whānau member. He is well respected and loved because he embraces each of our values in terms of respect, social accountability. Actually, I wrote them on my hand so I wouldn't forget them. Integrity and leadership. And I think Logan is also, he's our po manawa because he is so humble. And not long after I was assigned to be dean, and it was a steep learning curve for me, myself and my deputy dean did something really silly. We did a presentation to the researchers that if I could rewind and do anything different, it would be that talk that we talked to them about the current financial situation. And it wasn't done right. And instead of coming and saying, oh my gosh, what the hell were you thinking? Logan just gently came into the room. He didn't bring chocolate, that would have improved the experience, but he just softly spoke through some of the data points that I wasn't aware of. And it was so gentle and kind, and it was one of the best coaching moments I've ever had in my academic career. And I think it's some of the qualities that we really admire about your whānau member. And so thank you for sharing him with us. I must admit, as he was doing his presentation, all I could think about is medical research is such a complicated space. And it seriously is like the Hunger Games. And it's so competitive, and people lose their jobs, and we lose some amazing people, and we're glad that Logan got to come back and that we get to keep him, and that he's able to serve in such a great leadership role where he really is a kaitiaki. He really does look after those that are also in the vulnerable situation. So thank you for the values and the things that you've taught him, because we get to benefit from that. And I just wanted to say on behalf of all our staff that are here, thank you. And thank you to Logan, because he is so humble. I'm sure that he comes home and just goes, no. So I just wanted you to know the impact and the effect that he has on us daily. So thank you for sharing him with us. And with that, we get to now stand and do our last acknowledgement. I'll tell you what, Logan is going to be so glad when this is over, because he does not like attention. But if I can ask you to stand as our last bit of tribute to our the only person on our campus this year that has been bestowed the privilege of professor. So this is a really big deal for our campus too, that Logan has received this. And I asked Logan to lead us out along with his academic party, and then asked you all to join us for a kai and kōrero outside. Kia ora, Logan.